Hello, my name is Prashant Emani, and I'm an associate research scientist at Yale University, working with Professor Mark Gerstein. Today, I'm going to be talking to you about inferring identifying characteristics through the pooling of information across genotypic trajectories. Now, the basic premise of this project arose from the question of what is the degree of information leakage associated with even a small, sparsely distributed, and potentially noisy set of common genetic variants. This question in turn arose from a thought experiment we conceived where we imagined meeting an individual and trying to infer genetic variants, either based on certain external physical characteristics, on casual conversations and gleaning information about food allergies, for example, or even surreptitiously trying to gather DNA samples from contacted objects. Now, this information may seem innocuous, but the question is, can such limited genetic variation actually help to reveal identity, relatedness, or even socially stigmatizing phenotypes? Now, the very fact that this is possible arises from the fact that because of shared ancestry, or more broadly because of convergent genetic variation, individuals could potentially be cross-referenced against the increasingly large publicly available genetic databases. So the sorts of risks we're considering as a result of this are identity, for example, where you just say you were given access, you're able to gain access to a psychiatric disorder database. Can the inferred variation that you glean in that fashion enable linkage of individuals to individuals in that database? That same question could be asked about relatives. Can certain individuals' relatives be found in databases and therefore expose the individual themselves to exposure regarding that specific phenotype? There's also the case of the Golden State Killer, who was actually identified through DNA samples that were matched to those of relatives in ancestry databases. Finally, if all you had were partial or regional matches of an individual to others in the database, the question is, can the genetic risk of certain polygenic disorders be inferred? So our approach to assessing this degree of leakage was inspired by genomic imputation methods. Now, these methods are essentially used to try and fill in the gaps between genotyped loci using a reference panel of phased genotypes, for example, the 1,000 Genomes database, which we're going to use in the next few slides. So the idea is that you are going to try and find the most likely genotype at a given locus. And very often this is done using certain hidden Markov models. Now, we try to exploit a similar strategy, but instead of identifying the most likely SNP or the most likely genotype at a given locus, we actually calculate the best fit set of equally likely piecewise matches amongst the reference panel haplotypes. And hopefully, as we go on, each of those components would be highlighted and could be better understood. So this is a flowchart of the overall process, including our specific contribution. The idea is that you input these inferred or sampled genotypes, as well as a panel of phased reference genotypes that are very often publicly available. You feed these in to a hidden Markov model. And in this case, we use a very frequently used approach, a model by Lee and Stevens from 2003, where the latent space of the hidden Markov model is essentially a set of pairs of haplotypes. Precisely because we all have diploid genomes, the maternal and paternal haplotypes can be explored in a state space of pairs of haplotypes. Additionally, exchanges between these different haplotypes can be carried out through a Poisson process recombination formulation. And finally, there are mutation or genotyping errors that can be incorporated through this framework. Now, our specific contribution we have labeled PLIGHT, which stands for privacy leakage through inference across genotypic HMM trajectories. The idea is that we use a standard Viterbi algorithm, but also account for the fact that these are potentially sparse SNPs, and therefore incorporate all equally likely paths through this haplotype space. We also allow for flexibility in terms of the recombination model considered. For example, we can incorporate hotspots to given locations if they occur in between adjacent observed loci. We also allow for the ability to look at suboptimal paths, either because you're testing for robustness of your fit process, or even if you want to account for the fact that there was a lot of inherent noise in your input data set, and so you wanted to even consider some of the suboptimal paths, and so we allow for that. Additionally, we also incorporate a whole bunch of modules for downstream analyses, including visualization of parallel trajectories, the construction of these piecewise or mosaic genomes, but also, and importantly, we allow for the pooling of information across parallel haplotype trajectories, as I'll explain in a second. So first, let's start with the basic Lee-Stevens HMM model. Here, the observed genotypes up to, say, capital L loci form the set of observations with the maternal and paternal haplotypes forming the latent space. And of course, we explore the reference panel for all possible combinations of maternal and paternal haplotypes amongst the reference panels that best match to the observed genotype. We also allow for ancestral recombination so that you can actually have transitions from one haplotype to another, as well as mutations or genotyping error. Now, I won't address this in great detail, but basically the formulation is in the form of this sort of Bayesian product between a term that encodes mutation and a term that encodes recombination. Overall, this addresses the question, what is the probability of observing a set of genotypes conditional on an underlying panel of haplotypes? So the Viterbi algorithm that we employ in PLIGHT basically consists of trying to search through the space of haplotypes and looks for sort of these maternal and paternal paths through the haplotype space and finds the best fit pair of haplotypes to a given genomic segment. In this case, arbitrarily labeled haplotype 1 and 4 transition to labels to haplotypes 2 and 3 over another segment, which in turn transitions to 2 and N in a final segment of that chromosome. So we're going to search the entire space, incorporate recombination, incorporate mutation, and finally come up with a series of piecewise matches of reference haplotypes or pairs of haplotypes to the observed genotype. Now, it's important to keep in mind that given the fact that you have sparse data, the argmax function of this Bayesian product in the Viterbi algorithm could potentially produce multiple equally likely paths. Now, we term these paths genotypic trajectories just to capture the notion that what's happening here is a sequential exploration of haplotype space. So the net result is basically a series of parallel trajectories, all of which are equally likely to match with the observations. An important aspect of this is the pooling aspect, which is in the next slide, but also the fact that in any given genomic segment, the number of independent trajectories that are equally likely gives a sense of what we call genotypic entropy. That is, it gives a sense of how well constrained the genomic identity of a query individual is, conditional on the observations. So the pooling strategy, which like I emphasized before is important, essentially is best exemplified through the figure shown over here, where what we do is we try to calculate the polygenic risk score across all of the predicted haplotypes from our algorithm in segment 1 from the previous slide. So what I've done, shown over here, is a set of known risk alleles that are identified down below. And I find amongst all of the predicted haplotypes over here, we find the PRS scores for each of those independently, and then plot those against the overall population distribution of PRS scores. The idea being that if all of those haplotypes that are predicted for a query individual are in a statistically significant fashion found on one extreme or the other, it then says something about the risk to the query individual as well of those given phenotypes. So we're trying to maximize the information usage across all of these trajectories. So in addition to that, we also have certain memory bottlenecks. The basically in Stevens HMM basically has to deal with pairs of haplotypes. And so that basically increases the search space by a large degree. Now, we have an exact module which runs that same HMM exactly, but in addition, we also deal with a truncated module, which uses, similar to the Eagle imputation pipeline, only considers those trajectories that are within a certain margin of absolute optimality, and only those are extended from locus to locus. There's also an iterative module, which is somewhat approximate, yet is highly scalable, where what we do is essentially chunk up the problem into much smaller sets of exact problems, get the best fit haplotypes from those, pull them together, and then reinitialize the process and run it several times, and then pull that together to check for robustness of the final results. So armed with those, we then tackled certain example data sets. We have a pair of synthetic data sets where, in the simpler case where an individual is known to be entirely in a database, that is, the entire genome of an individual is in the database, you can randomly subsample 1,000 genomes individuals genotypes. We chose up to about 40 SNPs, mainly amongst common SNPs, because we wanted to capture the degree of leakage associated with common SNPs. And then we allow for varying mutation rates, so that from 0 to 30% mutation rates. For the tougher problem of individuals that are not known to be within the database, what we do is we randomly select 2 individuals and splice a given chromosome, half of which comes from one individual, another half which comes from another, with the possibility of then sprinkling some mutations on top of that. Finally, we do a test using sort of a left out set of relatives matched against the main phase 3 cohort of 1,000 genomes, and we present those results as well. So the results for the case of individuals known to be in the database were tested for just a single chromosome to keep things simple, where we average over 10 iterations and we run the number of SNPs from 1 through 40. And then we identify what is the minimum number of SNPs required to correctly identify a given individual. What can be seen from this is that up to a certain modest degree of mutation, about 0.1, you see that just 10 SNPs are enough to identify an individual correctly. As you increase the mutation rates, both the mean as well as the standard deviation of the number of SNPs needed increases. But this analysis told us that the identification seems to be remarkably tolerant to modest amounts of mutation or error rates. Now, it also tells us that the number of SNPs that are required to robustly identify individuals is very few. We next moved on to the another example. This is a simpler version of the pooling strategy that I mentioned earlier, but is somewhat illuminating because we used from the previous step of the individual known to be in the database certain examples, in this case with 11 SNPs. We found the predicted individuals, which did not include the ground truth query individual, but we compared the PRS score calculated in the manner shown over here between the ground truth individual and the non-ground truth predicted individuals. Now, for this analysis, the results weren't particularly remarkable because we found that while there were shared phenotypes amongst the top-ranked PRSs, none of them were truly statistically significant against the background. But we do believe that the strategy as a whole is potentially promising. Finally, we talk a little bit about the individual not being in the database. So as I mentioned earlier, we have these spliced chromosome examples over here, where in this case, 360 and 342 were chosen for half and half. And we find that chromosome 1, when searching amongst just 400 haplotypes in the exact algorithm, chromosome 1 does actually an excellent job of finding the true query individuals. Chromosome 2, on the other hand, has a little more variation where there's more equally likely paths, but one of the individuals is clearly identified. The net result is that cross-chromosome comparisons could be extremely useful in this analysis to truly identify the underlying individual. We wanted to scale that up to the full 5,008 haplotype cohort of 1,000 genomes phase 3. So what we did is for 20 random initializations, we ran this, and we found that chromosome 1 in this instance did not find both the underlying true individuals, but did find one of them. However, to emphasize this notion that cross-chromosome comparisons are important, when we compare it to chromosome 2, we find the other individual pops up, albeit with several parallel trajectories as well. Again, the notion of cross-chromosome comparisons is very powerful. So finally, we talk a little bit about the relatedness, where we try to look for first, that is parent, sibling, or child relatives, as well as second or third order relatives in a left out set of individuals for 1,000 genomes phase 3. We found one second order individual actually, but mostly we found quite a few siblings, parents, or children, signifying that there is a clearly identifiable risk that our tool is able to pick up on. To look forward to sort of new and further directions to consider, we thought that FastLS, a program by Gerten Lunter, which incorporates the positional Burroughs-Wheeler transform, was a very elegant and promising approach with great scaling properties. However, we weren't able to implement it in our case because there are no position-specific recombination rates allowed in the model. And that doesn't necessarily work for sparsely, unevenly distributed data. Next, we want to actually very easily transform this, not just into a SNP level, but also into a gene level analysis. For example, certain Mendelian disorders are known to affect entire gene loci, and that may be relevant. And that should be trivial for us to incorporate. Finally, and I'm happy to answer this in the question and answer session, but the possible usage in basic sample identity matching could be important. I want to acknowledge the contributions of Dr. Mark Gerstein, Dr. Gamze Gursay, and all of my other lab members, as well as my funding resources. And I want to thank all of you for your time. Happy to answer any questions.